So today we're reviewing units 2 and 3. Let's take a look at unit 2, the biological basis of behavior, meaning the brain and the body. Okay, 8 to 10% of the AP exams, I would expect of the 100 questions, 8 to 10 of them to be about this stuff. So we're going to go through the standards. For this particular unit, there were 9 standards. The College Board said, teachers, make sure you're teaching your kids. So the first 2 standards that the College Board gives us are the parts of a neuron and then how neurons communicate with each other. Do y'all remember us talking about neurons way back in the day? Okay, let's take a look. You probably have something that looks like this in your notebook. True or false? Great. So you need this information in your brain. All right. So neurons, and you have 100 billion of them in your brain. It's how you communicate. It's why you are the way you are. We start off with the cell body or the soma, the big part where the nucleus is. You have dendrites, which are these, they always say, finger-like extensions off the soma that receive information from other neurons. You have the long, skinny part that's called the axon. The axon is wrapped in a mushy, marshmallow-like protection called the myelin sheath. This word myelin or myelin sheath, it reminds me of the word marshmallow. It's like this white, fluffy insulation that helps speed the neural signal up when it goes down the axon. All right. At the very end, you have what's called the terminal, the end. Sometimes it's called the terminal buds or the terminal buttons. And that's where the neuron releases chemicals into the synapse. Do 2 neurons ever touch? No. There's always a little bit of space between the neurons. That space is called the synapse. Questions about standard 1, the parts of a neuron. Anything up here that you're like, I don't know what that is, Mr. Jones? Could you explain it real quick, just in case? Yes, Sarah? I mean, this is the neuron. The electrical impulse, the action potential goes from the soma to the terminal. So when the dendrite receives a signal, that message, it creates a little spark as those, on the mock exam, those the sodium potassium ions, they rush in and they create electrical spark and action potential that travels down the neuron. And when it hits the terminal buds, terminal buttons, that makes the neuron sort of spit out little chemicals into the synapse. Any other terms up here that you're like, Mr. Jones, I don't remember what that is. I'd love for you to go over it. Cool. So what are the nodes of Ranvier? By not asking me what that phrase was, I just assume that you all know what it means. So what are the nodes of Ranvier? Tell me. It's on the axon, kind of. What are the nodes of Ranvier? There are spaces in between the myelin sheath where those spaces are. That's a node, that's a node, that's a node, and that's a node. Okay. So make sure y'all know that because they could ask you on the AP exam to label a neuron, or they could ask you specific questions of the parts. Make sure you are aware of that. Okay. Standard 2 of 9. They want you to make sure that you know how neurons communicate with each other. So I have 2 diagrams for you. Here's my first diagram. This is what happens when that action potential gets to the terminal bud or the terminal button. Okay. So up here, this represents the axon terminal, the end of one neuron. And you have these synaptic vesicles, these little bubbles that the chemicals live in. Chemicals like serotonin and dopamine and acetylcholine, glutamate and GABA. Okay. They live in the synaptic vesicles. When the spark hits the terminal, when the action potential comes down the axon to the terminal bud, these little bubbles release chemicals into the space, the synaptic cleft or the synapse. And then those chemicals, if they're doing their job, they bind to receptors on the postsynaptic neuron, the next neuron, the neuron after the synapse, right? So this is where this new neuron, it is receiving the dendrites, I told you all, the finger-like extensions that receive signals. This is a dendrite on a new neuron. When it receives these little chemicals, it knows that now it's his turn to fire, to have an action potential, to make things happen. This is how neurons communicate to each other. You have 100 billion neurons in your brain. They fire something like 100 times a second. So constantly as you go through your life, neurons are firing. And that's why you do everything that you do. If you want to move your arm, if you want to think a happy thought, if you're trying to remember something, if you're trying to hit a baseball, millions of neurons in your brain are firing to tell your body how to do those things. Hopefully you remember seeing this graph of when a neuron fires. Neurons are normally in a negative state. All those charged ions waiting to go through their gates. Normally neurons are at negative 70 millivolts. When a neuron fires, when it depolarizes, when it becomes positive, we call that the action potential. Can a neuron immediately fire again? No. If you have your notebooks and you're trying to follow along on this page, you should have written down and put a star by a term called the refractory period. Make sure it's circled or starred or underlined. The refractory period is those little, the millisecond or two where a neuron cannot yet fire again. It has to get back to its polarized resting state before it can fire again. Questions about standard 2, how neurons communicate with each other? We spent an entire day talking about that stuff. I just tried to review it in 2 minutes. Make sure you're going back through your notes. Ask questions if you have questions, okay? So what happens when you take some sort of psychoactive drug? Well, those drugs can do a few different things. Some drugs can block reuptake, meaning when the chemicals get released into the synapse and they make the next neuron fire, instead of going back to their home, that's what reuptake is, some chemicals will block reuptake so that the chemicals, the neurotransmitters, keep bouncing around the synapse, making neurons fire. That's one way drugs can impact your brain. A second way is drugs can be agonists. If you look at the middle picture on the right, an agonist is a chemical that looks like and operates like a neurotransmitter. So it makes neurons fire more. This says enhanced cellular activity. Neurons fire more because of agonists. You can also have drugs that are blockers, antagonists. If someone has schizophrenia, what's the problem? What chemical is causing the problem for them? They have too much dopamine. They would take an antagonist like Risperdal or Thorazine for dopamine. And what antagonists do is they prevent the natural neurotransmitter chemical from binding to the dendrites of the next neuron so that these neurons can't and won't fire. That's what drugs do to your brain. They either make your brain fire more, make your brain fire less. Questions about how drugs impact neurotransmitters? Great, so depression. Serotonin is too low. I might give you an agonist for serotonin. So you don't just have the little serotonin molecules floating in the synapse, but you also have this drug that can do the same thing that serotonin is supposed to do. Great point. Does that make sense? Any other questions? Reuptake is that normally after the chemical does its job, the chemical goes back to its home. That's what reuptake is. So if you take a reuptake inhibitor, so the most famous one is an SSRI, reuptake inhibitor, it prevents the chemicals from going back to their home. The chemicals keep bouncing around and doing their job. It does the same thing an agonist does. SSRIs do the same thing agonists do. They make the neurons fire more. Someone's depressed, they have low serotonin and low norepinephrine. So you could give them an agonist for serotonin, or you could give them a serotonin reuptake inhibitor, and they would both do the same thing. Good point for depression. Either drug would be fine. The fourth and fifth standards that the College Board wants y'all to know from Unit 2. The endocrine system. What are some key words that we associate with the endocrine system? Hormones. Actually, that's the main one. That's really the one I want you to say. Thank you. Hormones are the chemicals in the blood that impact your body, especially around puberty. Testosterone and estrogen, right? Those sex hormones that impact the development of the gonads and the sex organs and sexual behavior. So the endocrine system shapes why you do what you do. And then the nervous system. This is standard 5. Okay, let me show you a diagram to help explain the nervous system. The nervous system, your nerves, meaning how your body communicates, how your brain communicates with the body. So within the nervous system, we have all these subdivisions. Do you have a chart that looks kind of like this somewhere in your notes? I hope you do. Try and find it. The mnemonic that I gave y'all was CPSAPS, CPAPS, and let's go over what exactly that means. So the C and the P, CP. Within the nervous system, we're either talking about the brain and spinal cord, the main stuff, the central nervous system. That's what's in blue over here on the right. If we're talking about everything else in your body that moves, that's your peripheral nervous system. Peripheral, meaning off to the side. So this is like the nerves that extend out to your knees and arms and legs and feet and fingers. Within the peripheral system, CP SAPS, CP SAPS, CP SAPS. Within the peripheral system, you have things that happen on their own. Reflexes, your stomach working, your lungs working, your heart operating, blood flowing throughout your body. There are some things that are just happening on your own that the brain's controlling. That's called the autonomic nervous system. And then there's some things that you have control over. Soma refers to body, right? The soma of the cell is the body of the cell. The somatic nervous system is when you are choosing to move your body. It's voluntary. You have control over these particular behaviors. So out of the things that you do not have control over, we can increase your brain's activity or lower your brain's activity. We can increase your body's arousal or lower your body's arousal. I've told you all year that the parasympathetic, it's like a parachute slowing you down. The parasympathetic slows you, relaxes you, calms you. Sympathetic, when you feel agitated and aroused and emotional, when there is a threat, the sympathetic nervous system is when there's more brain and body activity. You don't have control over either of these. They're just happening at all times, maintaining homeostasis within the body. What questions do you have about the way that the nervous system is structured? They love asking test questions like, Summer is getting attacked by a bear. As she turns to run, which part of the nervous system is active? We'd say the sympathetic nervous system is active. Yep. Nathan hits a tennis ball into the net. Which part of the nervous system is Nathan using? He's using the somatic nervous system. He is choosing how to operate his body, his muscles. Yeah, so he's only using the somatic system. These terms only apply to the stuff that's going on automatically you do not have control over. Your fear response, your digestion, your heartbeat, your sweat response. Those words are only used to describe things that happen on their own. If you can control it, it's the somatic system. All right, let's look at the next slide. The next standard the College Board gave us. We spent 2 whole days on the brain. Find that in your notes. What day was that? Unit 2, day 3. Okay. So we did 1 day where we talked about BFM Spavtal. We talked about the cerebrum. That's what's in this picture here. We talked about BFM Spavtal. What is stupid mnemonic? It's helpful for me though. BFM. B, Broca's area in the F, frontal lobe. The M, motor cortex, is also in the frontal lobe. BFM. Spavtal. S, sensory cortex, is in the parietal lobe. SP. O, occipital lobe, has the V, visual cortex. And the 4th lobe is the T, temporal lobe, where the auditory cortex, A, and the W, Wernicke's area are. BFM Spavtal. That's the cerebrum. That's the part of your brain in charge of thinking. That's what makes us human, okay? The outer part of the brain. So when you see terms like regions, lobes, cortical areas, it's referring to this stuff. Hemispheres. Which hemisphere is in charge of language? The left one does. It also does logic, math, school stuff. What does the right hemisphere do then? Creative art, music, things like that. All right. Now, if I could peel under this part of the brain, you'd see a ton of other structures. I don't have a graphic, but you'll have them in your notes somewhere. You remember any of the other structures of the brain? Like the 3 parts of the brainstem, or the parts of the brain in charge of memory, emotion, metabolism, the sensory switchboard operator. Do y'all remember any of those words? Shout some of them out. Medulla is in charge of heartbeat and breathing. Autonomic. Pons is in charge of dreaming and sleep. Cerebellum is in charge of balance and movement. The thalamus is the sensory switchboard. The reticular formation is in charge of how aroused, how awake you are. What's in charge of memory? The hippocampus. What's the brain's emotional center? Starts with an L, it's a system. The limbic system, limbo, it's in charge of your emotions. What's the part of the limbic system that makes you feel angry or mad? It's the walnut shaped. The amygdala makes you angry or mad. Am I missing any that you see in your notes? We got most of them. Make sure you remember the parts of the brain. Neuroplasticity, they love to ask about the fact that the brain can kind of heal itself if you have a traumatic brain injury over time. Is it better if you get a brain injury to be really young or older? When is your brain most plastic? When can it heal itself best? When you're younger. Yeah, that's what neuroplasticity means. Question? The medulla, it's in charge of the autonomic system of breathing and heart rate. Cerebellum, balance and movement. To be the belle of the ball, you got to have good balance and movement, hand-eye coordination, riding a bike, procedural memories, muscle memories. That's all the cerebellum. All right, something else we talked about in Unit 2 about the brain, the split brain research. Take a look at this picture on the right. It's pretty crazy. We've talked about why someone would have to have a split brain procedure, right? Seizures, epilepsy, too much electrical activity. So what's going on with this guy? What two words are flashed in front of him? The words key and the word ring. What is he going to say with his left hemisphere? What is his left hemisphere seeing? What word? Because the left hemisphere sees stuff to the right. So he's going to say the word ring. But his left hand is being controlled by the right hemisphere. And because he's a split brain patient, his right brain saw, what word was on the left? The word key. So with his left hand, he would pick up a key. He'd say, this is the word I saw. Out loud, he would say the word ring. But with his left hand, he would pick up a key. Pretty wild, pretty crazy. So that's Sperry and Gazzaniga's research. We talked about HM, who had memory issues with the hippocampus. We talked about how we can measure different brain activity. MRIs and CAT scans do brain structure. PETs and fMRIs do brain activity. You can do electrical stimulation of the brain to make your brain feel a little better. We talked about all these things. It's all in your notes, but let me know if you have questions. Unit 8 is stuff we've been talking about all year long. You talked about in biology class, nature versus nurture. Your genes versus your upbringing. The importance of genetics, natural selections, which traits survive. All right, what helps you reproduce the most? That those are the things that get passed on from generation to generation. Here are the only 5 names. Last unit, there were 13 names I had to put up on the board for you for Unit 1. Only 5 names for Unit 2. Paul Broca did what? What did Paul Broca discover? The part of the brain that's in charge of speaking, Broca's area. If your Broca is broke, your Boca no worka. Oof, rough. Wernicke's is hearing language, understanding, comprehending. Yeah, they work together. Broca's in the frontal lobe, Wernicke's in the temporal lobe. Darwin, obviously, evolutionary psychology, genetics, natural selection, blah, blah, blah. Sperry and Gazzaniga, these names always go together. They did the, what did Sperry do? They did the split brain research that we just looked at the picture of, right? The left hand, what you say. Wernicke, here's that word. Broca was speaking, Wernicke was understanding language. Okay, look through Unit 2 notebooks. B-Day folks who might be watching this, look through Unit 2 notebooks. If you see words or terms you want me to go over, send me an email or save them for class and we can go over them on Thursday for y'all, okay? These are the types of questions you should be able to answer from Unit 2, just like on your own automatically without looking at your notes. And this is the type of free response you might see for Unit 2. I'm not going to go over this one in depth and detail. I think you've seen it before, but the link will be on eClass if you want to kind of look at this. But basically, the types of things in the free response would be you understanding the different types of chemicals and neurotransmitters. So review those, how the nervous system is set up, brain structure, the different parts of the brain, the different things. So for this one, what part of the brain is in charge of your eating habits? Deals with your metabolism, not the thalamus. You got to get what? You got to get hype for the hypothalamus. So you would have to talk about the lateral and the ventromedial hypothalamus to get points for this free response. You could have talked about how there's an obesity gene, or how hyperthyroidism or diabetes can be inherited from your parents. And you would have had to talk about how some of those chemicals, serotonin makes you feel full. We talk about PYY and leptin and orexin. Those all impact your levels of hunger. So you may want to look back at those chemicals, those notes. But that's what you would have gotten the points here for response on eating and body weight. We're going to take a quick pause and then come back to look at Unit 3. We had a question about sleep because it was on the progress check for Unit 2. So the way sleep works is when you're super awake and focused, your brain produces beta waves, like beta club, super focused, good at school, paying attention, you're with it, you're awake, you're alert. When you start to relax, you go from beta to alpha. Alpha, still awake, but relaxed and chill. When you start to fall asleep, stage 1, those are called theta waves, okay? So you start to show theta waves in stage 1, and then as you get into deeper sleep, stages 2 and 3, those are where the delta waves, delta deep, okay? It's hard to wake you up from delta sleep. Remember in stage 2, on that mock, it asked about sleep spindles. So that's in stage 2. It's when your brain is starting to fall asleep, starting to get into deeper sleep. So it kind of freaks out a little bit. Now, the one thing people always forget about sleep is that you don't go stage like 1, 2, 3, and then REM. You go from stage 1 to 2 to 3, then you go back up to 2 to 1, and then you hit REM. So you don't hit REM in the first like 89 minutes of your sleep. The first hour and 20 some minutes, you're just in stages 1, 2, 3, 2, 1. Around the 90-minute mark, you fall into REM. This is when your body freezes up. It's like your body's chance to take a break. And your brain goes crazy. It looks like you're awake. Your eyes are rapidly moving, REM, rapid eye movement. This is when you dream. This is when you have nightmares, okay? And those last for, 20 minutes or so. Then you go back into stage 1, 2, 3, 2, 1, and you'll have another REM. As the night goes on, your REMs actually get longer and longer because you're spending more time dreaming and you're not spending as much time in that deeper stage 3 sleep. So as the night goes on, you dream more. Night terrors are not the same thing as nightmares. Nightmares are just bad dreams. Night terrors happen when you're in the deep sleep. And that's when you can like get up and walk around the house and scream and freak out and scare people, and you won't remember any of it the next day. All right, let's talk about Unit 3, sensation and perception. It might be the smallest unit of the entire year. It's only 6 to 8%, but there's a lot of complicated content that I know y'all have forgotten about the senses. So let's go ahead and dive on in. The first thing we did, and in your notebooks, follow along, Unit 3, probably day 1 or day 2, we talked about how your brain detects signals, whether you can even notice if there is a light, a smell, a sound, something touching you. So that's called an absolute threshold. It's the minimum stimulus you can detect 50% of the time. The smallest possible sound or taste or smell or light. Now, if you sense something and it's changing, that's no longer your absolute threshold. It's your JND, your just noticeable difference. The smallest change that you can detect about half the time. We all have different JNDs. So if there are 4 people in my room who are cold right now, if I bump the temperature up 2 degrees, it's possible that one of them now thinks it's too warm. One of them thinks the temperature is perfect. And 2 of them can't even tell that it changed because we all have different JNDs. That's the threshold that we notice in terms of change. If you see the phrase signal detection theory, the example I gave y'all constantly was about there being a bear in the woods and whether or not you correctly noticed a bear in the woods. If there is a bear and you act like there's a bear, we call that a, hey, good job. There is a bear, your response is like there's a bear. Good job. If there's no bear and you don't act like there's a bear, also good job. It's in green down here, bottom right, correct rejection. What if there's a bear and you don't act like there's a bear? That's a miss. What if there's no bear and you act like there's a bear? False alarm. What's the worst of these according to researchers? And out of all the scenarios I just gave you, what's the worst possible scenario? This is what your brain tries to prevent. This is why, side theory from Mr. Jones, this is why humans are so anxious. We are constantly going through life as if there were threats, as if there were bears, as if there were things that could harm us, as if we were being at risk of being kicked out of our social group and starving. This is why we love eating ice cream and why we latch on to our groups and our cultures. It's because we would rather have a false alarm. We'd rather respond to a threat that's not actually there than potentially miss a threat that could hurt us. So our brains, bless you, are overactive because we're trying to avoid misses. Now, as you go through life, you're exposed to various stimuli. We have sensory adaptation and habituation. These words mean different things. Similar. Both of them are your brain and body getting used to some sort of stimulus, but adaptation is your brain getting used to a constant stimulus, like you jump into water that's a little bit cold, or there's a buzzing sound in the background. That's sensory adaptation. Habituation is the train example. The train goes by so many times a day that like you don't notice it doesn't scare you the way it did at first. When you're using a drug for the first time, it freaks your body and brain out. But over time, you build up a tolerance to that drug. So the same amount of drug, it doesn't have as large an effect on you as it used to. That is a form of habituation. It's how your body gets used to a particular substance or stimulus. Questions about anything here in standard 1? Standard 2, we're gonna do 7 slides on standard 2. Because this is all the stuff y'all have forgotten. It's all the different senses. So I got this beautiful picture of an ear. This is actually on that mock exam. We talked about how sound waves, vibrations in the air, travel into the ear canal. They bounce off the tympanic membrane, also known as the eardrum. They go off the ossicles, these 3 bones, the incus, malleus, and stapes, also known as the hammer, anvil, and stirrup. The sound waves go into the oval window, into the cochlea. The cochlea is filled with a fluid, hair cells that vibrate on the basilar membrane. And those vibrations send a particular signal to the brain up the cochlear nerve into the temporal lobe. And that's what tells your brain what sounds you hear. Are these purple loops part of hearing, the semicircular canals? Do those have to do with hearing? No, what do those have to do with? Balance. Very good. Any questions about the ear? You need to review frequency theory, pitch theory, why we hear the high sounds, the low sounds based on the vibration frequencies. Okay, you lose your high pitch sounds first. We talked about the eye. Light waves go through the cornea, the clear outer covering that you touch if you put on contacts. They go through the aqueous humor, this liquid here that gives your eyeball its shape. The light waves pass through the lens, through the vitreous humor, all this liquid in the posterior chamber, and those light waves travel through this liquid, and they hopefully will focus on the back of your retina. It's that tissue layer with rods and cones on the back of the eyeball. The fovea is where you have the most cones. You see the most color, you see the most acuity, detail. I was trying to make sure that was right. The foveas, that was just like me thinking. The fovea is where you focus on details and color, acuity, clarity. That's the fovea. You have a blind spot. There's a point here on the retina where you actually can't see anything because there's no rods and cones there, because that's where the optic nerve connects to the retina. And then as you go further along the retina, you have less cones, so that in your peripheral vision, there's that word peripheral again, off to the side, you can only see black and white and gray. So your rods off to the side, black and white, are in charge of your nighttime vision, whereas cones operate when you're in the light, and cones can see color. Questions about the eyeball and vision? Cones are the ones that see color details, yes. Trichromats, dichromats, opponent process theory, how colors have opposites. Make sure you remember all that stuff. The trichromatic, the Helmholtz theory for hearing. Nighttime, rods do nighttime vision, black and white, gray, outline shapes of things. Taste, also called gustation. You have papillae, taste buds on your tongue, and that's what allows you to taste the 5 different flavors. What are they? Sweet, sour, bitter.
Savory and umami.
Is savory one? Umami. I know umami is the one they love to ask about. Savory? We think it's savory? Cool. Is that the same thing as salty? Maybe. It's a combination. All right. What's the fancy name? If gustation is the fancy word for taste, what's the fancy word for smell? It's like Olaf. Everyone say olfaction. Sometimes they ask about the olfactory bulb, represented by this yellow image here. It's like a light bulb. It hangs down from the roof of your nose, your nasal cavity. The unique thing about taste and smell is that unlike hearing and sight, where you're interacting with light waves and particles, right? Vibrations. With smell and taste, you actually come in contact with a chemical. Whenever you taste something, the chemical is on your tongue. Whenever you smell something, the chemical has gotten into your nose. Why is smell the most powerful sense? It brings back memories. Why does it bring back memories? It is connected directly, hardwired into the brain, into the limbic system. It's the only sense that does not go through the thalamus, the sensory switchboard. Absolutely. That's why smell is so powerful and why it can bring back memories. Vestibular and kinesthetic senses. This is still part of standard 2 from unit 3. Vestibular is your balance, so semicircular canals. Your cerebellum works with your vestibular sense. Kinesthetic sense is about your ability to move individual body parts and to know where your elbows and where your knees and where your feet are. Have you ever had to walk through a crowded, cluttered room before without trying to do it without touching anybody? Right? Without touching anything, without bumping anything? That's you using your kinesthetic sense. You're literally moving and you're very aware of where all your body parts are. Are any of you clumsy? You're always knocking things over and dropping things? If you are, that's a problem with your kinesthetic sense. It's about your body's movement and where specific body parts are. So the kinesthetic sensors are in your muscles and your joints, where your bones connect, so that your brain can be aware of where your body is at any point in time. If you have a good kinesthetic sense, we do this activity where we try to get you to match your fingertips up perfectly. That's you having a good kinesthetic sense. The last sense from standard 2 is about pain. This diagram is kind of hard to see, but it's representing the gate control theory of pain. Remember, you have multiple types of pain fibers. You have the A+, A delta thick fibers that communicate emergency bad fast pain to the brain. And you also have the smaller B and C and D fibers that don't carry really important signals. It's like less important, soreness, throbbing pains, aches that you might have. And the idea of the gate control theory is that the spinal cord, only one message can get through at a time. So if we can manipulate your nerves into sending other messages to the brain, then maybe that bad pain won't get through. That's what acupuncture and some massages and some other forms of therapy do, trying to prevent the pain signal from getting up to your brain. Questions about the senses? All right, let's talk about things that can go wrong. Synesthesia. They added this to the College Board standards, so they might ask about it. Synesthesia, and I don't even know who talked about it. Synesthesia is when your senses are not doing their job. You want to write that term down. Some people with really weird instances of synesthesia, when they hear certain sounds, other senses activate. So when they hear a sound, they might see a certain color or taste something. There's really rare cases where people feel something or they smell something. In other cases, the opposite. When they see certain people, they have certain sounds that play in their mind. It's like the wiring in your brain related to the senses has gotten all mixed up, so the wrong senses activate. The most famous instances are it's sound related to color. So certain words lead to certain colors, and therefore every word has a unique shade of color that they just see in their mind. It can be very distracting, but they also say it's very beautiful. Question?
Does that have anything to do with schizophrenia?
Does it have to do with schizophrenia?
Or does it even matter if I know that or not? Do you think?
I bet it probably doesn't matter if you know that. I bet there's some overlap, right? Because it's kind of like having hallucinations almost, right? Hallucinations are when you sense things that no one else can sense. So I'd say it's kind of related, but it's probably its own thing, and it doesn't come with all the other stuff that schizophrenia has. Yeah. So we talked about some of these other impairments. Prosopagnosia, they love to ask about. I have it bolded here. Face blindness, when you have trouble recognizing people. We talked about the ventral and dorsal streams. People who have those messed up. Ventral is what is the object. Dorsal is where is the object. We talked about the man who mistook his wife for a hat. Those are like forms of synesthesia and sense problems. Those of us who wear glasses or contacts, we might be myopic, meaning we can only see things that are close by. We are nearsighted. Hyperopia, you can see things that are far away. Astigmatism is when your lens is shaped weird, and that can impact your vision. With color blindness, you can lose a single color pair. You can be red, green colorblind, which means instead of being a trichromat, like many of us, being able to see all three color pairs, black, white, red, green, blue, yellow, they can only see two of them. Some folks have really severe color blindness where they can only see one color pair, black, white. So chromats always refers to color. When we talk about hearing problems, two different types of hearing loss. It can be a physical, mechanical, like a bone eardrum problem, where the waves can't get to the cochlea. That's called conduction deafness. A hearing aid will amplify the amplitude of the waves, make the volume louder so the waves can still get to your cochlea. You can also have the more severe nerve deafness, where like the cochlea is messed up or the nerve going to the brain isn't carrying the signals properly. In that case, they basically have to replace your cochlea and the nerve in your brain. They have to rewire this machine, this device into your temporal lobe so you can hear sounds. That's more extreme. Make sure you know about like colors. The hue is determined by the color's frequency, and then the brightness is determined by the wave's amplitude. With sound, pitch is frequency, but volume is amplitude. Make sure you remember how waves work. Questions about standard 3? Gestalt laws, yeah, would be part of sensation and perception, illusions, things like that. That actually will be up here with this stuff. How your brain interprets. So this unit, sensation and perception, we've been talking the first 3 standards all about sensation. These last few standards are about perception. So how your brain takes all this information and what it does with it. Sees depth. There's one-eye depth cues and two-eye depth cues. It's like the one-eye depth cues. Which circle is closer to you, the blue one or the green one here on the very top left? Which one feels closer to your brain? Blue. Why? It's occluding, it's blocking the back one. Which shape seems closer to you, the one on the left or the one on the right? This one feels closer to you because it is bigger. The shadows, that's one way that you can tell depth, right? You're going to think that this object is further from you because it has a smaller shadow. That's interesting. Shading determines whether something is coming towards you or going away from you. How close something is on the horizon, that helps you out. Linear perspective, the fact that lines converge as they're further away from you. The amount of texture you see in something and the bumps, that tells you whether it's close or far from you. All these things are depth cues that only require one eye. There are binocular depth cues. There's only 2 of them. Convergence, the fact that your eyes literally curve inward when something's moving towards you. That's how you can get yourself cross-eyed as you follow your finger in towards your eyes, because your eyes, they go too far. Retinal disparity is the idea that each retina, each eye, sees a slightly different version of the world. And the reason you see in 3D, the reason why 3D movies work is because we can send a slightly different image to your left eye and right eye, and your brain just automatically combines those images. That's how your brain sees depth in 3D using both eyes. If you're blind in one eye, neither of these will work for you because your brain is not taking in information from 2 eyes to do the math on, to figure things out. Other stuff about how your brain deals with sensations and perceptions. Top down versus bottom up processing, meaning is it your brain starting with the idea or is your body collecting information about the world around you? Y'all know about attention. You can't multitask. They love asking about that. Divided attention is not effective. If you think you can listen to Mr. Jones and also be on your phone, you're wrong. You are not getting all the information, and it impacts your memory. It impacts the way your brain processes things. It's going to be a problem for you. So selective attention, you're only paying attention to certain things, but you're really focused on those things. Questions about anything as you're flipping through about sensations and perceptions so far in your notebooks? The word perceptual sets, it's what you expect something to be like. So I showed you this picture, and if you go from top to bottom, what's the symbol in the middle? A, B, C. But if you go from left to right, what's the symbol in the middle? 12, 13, 14. So perceptual set has to do with your expectations. You see what you expect to see. That's the idea. And that's why we can trick your brain with all those different illusions. You can go back and look at the different illusions, the Gestalt principles, and sometimes we're just wrong, y'all. We think, so the parapsychology, this idea that some people think that they can have ESP or they can move things with their mind or they can communicate with people who are far away. Telekinesis, moving things with your mind. ESP, seeing the future. What's that called? Seeing the future. Is it clairvoyance? Seeing the future? What's when you can see something that's somewhere else, that's going on elsewhere? All those like weird things that have not been proven by psychology, by science, those are called the parapsychological phenomena. Okay, thinking that you can have subliminal messages that you can read and that no one else can, that kind of stuff. Only 4 names from this unit: Weber and Fechner, who created the Weber-Fechner law. I call it the grocery cart law. I said, if you're trying to sneak something into the grocery cart, sneak something in when the grocery cart's really full, because your parents are less likely to notice it. The Weber-Fechner law is the idea that when there's a lot going on, it's going to take a bigger change for you to notice that something changed. If the volume's on 5, you might notice it going to 6. When the volume's on 30, going from 30 to 31 might not get noticed, because it's all about how big a percentage or a proportional change. Hubel and Weasel, do you remember what animal they worked with? They worked with cats. I showed you all that video of the cat. It got strapped down and it was looking at very specific symbols, and it was hooked up to very specific neurons, and those neurons only fired at very specific lines, edges, angles, curves. Those are the words that we had in that free response that you might see on the AP exam. So feature detectors is the idea that some neurons are very, very specific in terms of what they'll fire at, what will activate them, very specific features. Anything else y'all see in Unit 3 from your notebooks? So let me show y'all an actual free response from the College Board that had to do with Unit 3. I'm going to keep recording for a little bit. I want y'all to see this here. Okay. It was a weird prompt. I've never seen this format before. The prompt said, for each of the following pairs of terms, explain how the placement or location of the first influences the process indicated by the second. It's worded really weird, but basically saying, how did the location of rods impact peripheral vision? How does the placement of words on a list, how does that impact your ability to recall those words? How does the location of serotonin impact depression? How does the placement of your eyeballs affect depth perception? How does your motor cortex affect what your body does? How does the existence of other people around you, how does that impact your performance? And how does 2 things being close to each other impact the way you view those things? It's a really weird prompt. Let me show you. How many points would this prompt be worth? 7 points. What should your first words be? The location of rods. Where are rods? Are they in the center of the retina or are they on the edges of the retina? Cones are in the center on the fovea. Rods are on the edge. I'd say the location of rods on the edges of the retina allow you to see black and white shapes off to the side of your peripheral vision. That's how I would answer number 1. The location of rods impacts peripheral vision because it only allows you to see black and white and gray. You can't see color in your peripheral vision because the rods are the only thing on the edges of the retina. That's how I answer number one. Let's look at how an actual student responds to this prompt. For number one, they wrote, the rods which are lonely, that's so sad. The rods which are located mainly on the outer regions of the eye and are responsible for light dark help with peripheral vision because since they are on the outside of the eye, can detect images on either side. Did they do a good enough job of explaining what rods do to peripheral vision? Would you give them point one? Okay, let's take a look at point two. This is from unit 4 or 5. So like we haven't reviewed this yet, but maybe you just remember. Word recall is influenced by the placement of the words because their person is more likely to remember the words listed at the beginning and ending of the list and forget the ones in the middle. This person had terrible spelling. Did they do a good enough job of point two to explain how the location of words impacts recall? Do you think they got point two? Okay, let's give them a check. Number 3, people who lack the proper amount of serotonin will have depression. So increases the serotonin that the body has will reduce depression. The placement of serotonin in the synapse is important because drugs that are selective serotonin uptake will leave more serotonin in the synapse and thus reduce depression. Does this person have good grammar? No. Did they get enough right about serotonin to get point 3? A little confused, a little lost. Well, they said, are they right that more serotonin will reduce depression? If you think that phrase is true, then that's good enough to get the point. It seems to me like they weren't really sure what the prompt wanted from them. So they were like serotonin. So they like talked about like serotonin in the brain, in the body. And then they talked about serotonin in the synapse, like just in case. It seemed like they were covering their bases. That's a great thing for them to do. They got points 1, 2, and 3. Even though they had typos and bad spelling and grammar, they actually got all 3 of these points. Let's look at point 4 and 5. Retinal disparity is the process in which your 2 eyes see different images and your brain works to combine the 2 images. This relates to depth perception because when the images are closer, the 2 images have greater difference and the brain must work harder to combine them. They got point 4. They explained how this binocular depth cue works. Point 5, the motor cortex in the brain, BFM, in the frontal lobe has different size representative areas for different parts of the body, controls the movement of the body. Body movement that requires muscle movement is more largely represented in the motor cortex than other parts. The hands and face are overrepresented because there are more muscles to move. You remember the picture I showed y'all of Mr. Homunculus? He had the big lips and the big hands and the big fingers. So is what this person said true? Yeah. The motor cortex is in charge of controlling body movement. So they also got points 4 and 5. This is an actual student response. Let's look at point 6 and 7. The social facilitation effect. This is in our last unit of the year, where people perform better at tasks they are good at when others are around. So the presence of others being close to the person performing the action increases the performance of whatever is being displayed. Again, they misspelled performing, they misspelled increases, but did they correctly explain what social facilitation is? This person got point 6 and point 7. Proximity is how close something is to another thing. If objects are closer to each other, then the brain is more likely to see them as being grouped together. Emily asked about Gestalt. What do we know Gestalt stuff? And they kind of give an example here. This is the Gestalt theory. This student, maybe one of the worst writers I've ever seen in my entire life, they actually got all 7 points, the College Board said. They got perfect score on this free response. So y'all don't have to be perfect. You just have to communicate that you understand the concept. The more information you put, the better. Okay. So here's what the College Board said and why they got all 7 points. Even though they didn't use the exact right terms necessarily, they like still got the points because they did a good enough job of describing the ideas. Okay. Y'all need to have finished through progress check 3. There are daily videos you can watch if you want. Keep going back through your notebooks. B-Day folks, email me questions if you have questions. Zoomers, stick around if you want. I recommend that y'all retake these old tests. I've opened them up for you so you can take them as many times as you want. Test yourself. It'll show you which ones you missed and what the right answers were. Message me if you don't understand a question. Go back on eClass, y'all. Look at the guides to see the list of vocabulary. You can get on Quizlet. There are Kahoots. There are documents about the brain. You have your test corrections. You can rewatch the crash course videos, 3 through 7, cover today's content. So there's 5 crash course videos, 10-minute each, that would teach the same stuff. I went over it in a slightly different way. If you want to hear it from somebody else, y'all can rewatch the homework videos that I put on YouTube from early this year, 10-year accomplish Jones 21 YT, or y'all can get on my Quizlet and play games and things. Okay. B-Day, folks, do some studying. I'll see you guys Thursday.